Dear student, so today we are going to see some of the very important questions from the chapter cell cycle. Here we will mainly focus on the statement based assertion, reason and some number based question. This chapter is very important. So in this video, if you want to solve the question by yourself, just pause the question and you can get the answer by yourself. If you want to increase the speed, you can increase the speed to 1.5 and you can go through the questions very fast. So let's get started. The first question, as it says that, choose the correct event during the cell cycle. Genome duplication, yes, it happened during the cell cycle. Synthesis of cellular constituent means the cell organelle, yes, they are also synthesized. Cell growth and DNA replication is a continuous process. Now cell growth is a continuous process, but is DNA replication a continuous process? No, DNA replication is not a continuous process. DNA replication only take place in the S phase, so this is wrong. Then distribution of chromosome to daughter cell with intact genome, yes, this is also right. And then genetically controlled event, and it is a random sequence of event. Is it a random sequence of event? No, it is a coordinated sequence of event. So that's why both 3 and 5 are wrong. So 1, 2 and 4 is the correct answer. Let's quickly move to the next question. As it says, choose the correct statement. Here cell growth with respect to DNA replication is a continuous process. We know that cell growth is a continuous process, but with respect to DNA replication, no, the NCERT says that it is not DNA replication with respect to increase in cytoplasm. Right? So this is a wrong statement. Then during the cell cycle, the genome remain non-intact. Just we have seen the genome remain intact. So this is also wrong. During cell cycle, DNA replication take place in the daughter cell. Does it take place in the daughter cell? In daughter cell already replication happens. So this is also wrong. In daughter cell it doesn't happen. During the cell cycle, along with duplication of genome, other constituents are also synthesized. So 4 is the right answer here. 1, 2 and 3 are wrong here. Next question, which of the following is correct? The typical eukaryotic cell is illustrated by yeast cell in the culture. NCERT, read it. Here it is not yeast cell, it is by the animal cell. Yeast cell divide once in approximately 24 hours. This is also wrong. Human cell divide in 24 hours. Cell cycle is divided into 3 basic phases: interphase, karyokinesis, cytokinesis. Is it right? No, this is also wrong. It is divided only into 2 phases, interphase and M phase. NCERT line, it is divided into 2 phases. Interphase is the phase between 2 M phase. This is a correct statement. So 1, 2 and 3 is wrong. 4th one is the correct. Next question, which of the following is correct? In human cell, the interphase lasts for about an hour. Interphase lasts for about an hour? No, interphase is greater than 95%. So it lasts for more than 23 hours. So this is wrong. Interphase lasts more than 95% of the duration of cell cycle. This is correct. M phase lasts more than, no, it lasts less than 5%. Actual cell division take place in interphase. Actual cell division take place in interphase. This is also wrong. It take place during M phase. Next question. Which of the following is true about interphase? It is called as resting phase. Correct. Cell prepare for cell division. Correct. DNA replication occur in interphase. Cell growth occur in M phase. See, DNA replication occur in interphase is correct, but cell growth, it is a continuous process. So not only in M phase, it also take place in interphase. So this is incorrect. Then separation of daughter chromosome occur during cytokinesis. During cytokinesis, separation of daughters. So it is also wrong. During karyokinesis. During cytokinesis, cytoplasm is separated. So this is also wrong. So 1 and 2 is correct here. Next question. Consider the following statement regarding cell cycle. The amount of DNA in a cell double during S phase while chromosome number remain unchanged. Yes, right. 2C become 4C, but 2N remain 2N during S phase. Correct. Histone protein are synthesized only during G1 phase. This is wrong. Histone protein is synthesized during S phase. Then cell that do not divide further enter into an inactive stage called G0 phase from G1 phase. This is also correct. During G2 phase, the cell prepare for mitosis by synthesizing protein during protein required for spindle formation. Yes, this is also right. So except 2, all are correct. So 1, 3 and 4 is correct here. Next one, if we see, which of the following is true for G1 phase? G1 phase, it is the interval between 2 M phase. Is G1 phase interval between 2 M phase? No, it is true for interphase. Interphase is the interval between 2 M phase, not G1 phase. Cell do not grow continuously. During G1 phase, cell grows, right? So this is also wrong. Cell are metabolically active. Yes, in G1 phase, cells are metabolically active. DNA replication occur. This is also wrong, right? So only 3 is the right here. 1, 2 and 4 is wrong. Next, which of the following is true about S phase? So DNA synthesis take place. Yes. Number of chromosome double. Number of chromosome remain the same. Number of DNA molecule double. Yes. Number of chromosome remain the same. Correct. DNA content get double. This is also correct. So except 2, all are correct. So 1, 3, 4 and 5 is correct. Next one, which of the following don't take place in the S phase of the plant cell nucleus? S phase, then plant cell, and then in nucleus. 3 factors are here. So during S phase, DNA synthesis take place. It take place in plant and it also is in nucleus. Centriole duplication. In plant there is no centriole duplication. Protein synthesis. Now you will say in S phase protein is synthesized, but does it happen in nucleus? No. So it doesn't happen in nucleus. So first is the right answer over here. Next one, at the end of S phase, onion cell will have how many DNA molecules? See, onion cell in NCERT they have given number of chromosome. How many chromosome do onion cell have? So onion cell have 16 chromosome. So how many DNA? We know that during the end of S phase, each chromosome will have 2 chromatid. One chromatid is 1 DNA molecule. Right, so one chromosome has 2 DNA molecule. So 16 chromosome will have 32 DNA molecule. Understand it? So always remember, number of chromatid is equal to number of chromatids is equals to number of DNA molecule. Remember this. Next question. Let's see. If onion cell, again from onion cell, 12 picogram. Here it is the DNA content. 12 picogram at G1 phase. At the end of G2 phase, DNA content will be. See, we know that in G1 phase, if the DNA content is 2C, S phase, by the end of S phase, it becomes 4C, and in G2 phase, it is 4C. So if 2C is 12 picogram, 2C is 12 picogram. So in G2 phase, it will be 12 into 2, that is 24 picograms. So third is the right answer. Next one, onion cell have dash DNA molecule in G0 phase. Remember, in G0 phase, the number of chromosome remain 2n, right? So if number of chromosome is known, then the number of DNA molecule double of that. Now, because it is in G1, so understand, G0 is just after G1 phase, means there is no S phase. Right? So in G1 phase, each chromosome has only 1 chromatid. In S phase, each chromosome will have 2 chromatid. So here, what we will say, that 1 chromosome is equal to only 1 chromatid. So in G0 phase, the number of DNA molecule will remain 16, because during G0 phase, each chromosome has only 1 chromatid. Number of chromatid equal to number of DNA molecule. So very important question. So here, in this question, it is asking about G0 phase. So all cell must enter into G0 phase. No, some cell may not enter into G0 phase, like the cell which is continuously dividing, the cells of the root of the cells of the hair root. And then there are some cell which remain in G0 phase. Yes, there are some cell which remain in G0 phase, like nerve cell. So this is also wrong. It's called an inactive state. This is correct. Cells are metabolically inactive. This is wrong. Cells are metabolically active. Cell in this phase can proliferate. No, this is wrong. They cannot proliferate. So only 1 is correct. Only 3 is the right answer. Next one. Which of the following statement is true about G0 phase? Okay, this is a repetition. Fine. Next one. Choose the correct statement. All diploid cell undergo both mitosis and meiosis. See, when I say all diploid cell, now, it is wrong. All diploid cell will not undergo both mitosis and meiosis. Wrong. Mitosis always take place in diploid cell. This is also wrong. Mitosis may take place in haploid cell. Then male honeybee, mitosis take place in haploid cell. This is correct. Lower plant, mitosis take place in haploid cell. This is also correct. Mitosis mostly take place in haploid cell. Wrong. Mitosis mostly take place in diploid cell. Plants show mitosis in both haploid and diploid cell. This is correct. Yes. Then animals show mitosis only in diploid cell. This is also wrong. Animal cell may also show mitosis in haploid cell. So here 3, 4 and 6 is, so 6 will be the right answer. These 3 statements are correct. Let's move ahead. Which of the following is true about cell division with respect to mitosis? It is a regressive process. No, it is a progressive process. Clear-cut boundary present between different stages. No, there is no clear-cut boundary between different stages. Karyokinesis has 5 stages. Karyokinesis has 4 stages. Wrong. Number of DNA molecule in parents and progeny are the same. This is correct. right? During mitosis, number of DNA molecule in parent and progeny is the same. Then assertion region. Z0 phase is called quiescent phase, correct? Cell in this phase are metabolically active. So see, this is also correct. But because it is metabolically active, it is not called quiescent phase. Quiescent phase it is called because it is inactive phase, right? So that's why explanation is not correct. So A and R are true, but not it is not a correct explanation. You know that in G0 phase the cell do not divide. That's why it is called as quiescent phase, and that's why second is the right answer here. Next one, match the following. So we can see resting phase. So interphase is called as the resting phase. Inactive phase, G0 phase is the inactive phase, longest phase. G1 is the longest phase. Division phase, M phase is the division phase, and synthesis phase. During this phase, the histone protein is formed. So here the correct answer will be second, right? So second will be the correct answer here. Let's move to the next one. Choose the correct statement. First stage of karyokinesis is followed by S and G2 phase. See, karyokinesis, is it followed by S and G2? The answer is no. It is written opposite. S and G2 is followed by first phase of karyokinesis. So this is wrong. During S and G2 phase, DNA molecules are untangled. So during S and G2 phase, DNA molecules are untangled. They are entangled. So this is also wrong. The chromosome material are intertwined during the process of chromatin condensation. See, whenever there is a chromatin condensation, then the chromosome material get untangled, not intertwined. So this is also wrong. Remember, two terms here are important: untangled and intertwined. So this is also wrong. Prophase is marked by the initiation of condensation of chromosomes. So this is a correct statement. So this is a very important question. Just go through it. Next one, it is asking the completion of prophase. Completion of prophase means by the end of prophase. Initiation of chromosome. See, at the end there will not be a initiation of chromosome condensation. It is not asking general about prophase. It is asking about completion. So this is wrong. Centrosome begin to move toward opposite pole. Yeah, this is right. Condensed chromosome with two chromatid attach at the centromere. This is also correct. And formation of mitotic apparatus. This is also correct. So 2, 3 and 4 is the right answer. See, if you read NCERT, there are 2 points given in prophase. These 3 points are included in the second point. Next one. Which of the following are correct? So this is important related to the mitotic apparatus. So in this, each aster has 2 centriole. Each aster has 2 centriole. This is correct because see, each aster has one centrosome and one centrosome has 2 centriole. That's why each aster has 2 centriole. Each mitotic apparatus has 2 centrosome. Yes, this is also correct. Okay, let me draw quickly the structure of a mitotic apparatus. See, at 2 pole, these two centrioles are arranged. This centrosome along with the radiating microtubule, this is called aster. This is called aster. Now, same way on the other pole, the radiating microtubule along with the centrosome, this is called aster. So you can clearly say that if I say aster, so aster is centrosome plus radiating microtubule, right? And here, if you see, now if I add, let's say, using different color, if I show here, let's say this. So here they will form spindle fiber like this. So this is the spindle fiber. So now both the aster along with the spindle fiber, now this together it will be called as mitotic apparatus. So what we will call the mitotic apparatus? It is 2 aster plus spindle fiber. Spindle fiber. Now if we see this question, then we can answer it better. Now here if we see which of the following is correct. So each aster has 2 centriole. Yes, each aster has 1 centriole, another centriole. So it is correct. Each mitotic apparatus has 2 centrosome. See, each mitotic apparatus has 2 centrosome. This is 1 centrosome. This is another centrosome. So each mitotic apparatus has 2 centrosome. This is also correct. Aster doesn't have spindle fiber. Aster doesn't have a spindle fiber. This is also correct. See, aster is centrosome plus radiating microtubule. So where is the spindle fiber here? No. If I add spindle fiber to aster, then it becomes mitotic apparatus. So aster doesn't have spindle fiber. This is also correct. Each mitosis involve 4 centriole in the mitotic apparatus. Each mitosis involve 4 centriole. See, in this, isn't it 4 centriole? 1, 2, 3, 4. So this is also correct. So the correct answer is all of the above, means 1, 2, 3, 4. All are correct here. Next, let's see. So just here, this one, you need to go through it. Very important concept. Again, it is given in the last paragraph of prophase in NCERT. Then next one, if we see this question here, which of the following organelle didn't disappear during prophase? Now we know that during prophase cell organelle disappear, but which cell organelle don't disappear? See, Golgi complex, it disappear. ER disappear. Mitochondria, see, mitochondria don't disappear because ATP formation or ATP is required during cell cycle, so mitochondria do not disappear. Centriole doesn't disappear, it remain there. Nucleolus disappear, nuclear envelope disappear. So third and 4 will not disappear. So correct answer is 3 and 4. Next one, in this question we have to arrange the events in a correct sequence. So this is related to mitosis. So basically here first we will see what are the stages of prophase. So here we see formation of aster, this will be the first one. Then next will be, so this is the first one, formation of aster. Then second one will be complete disintegration of nuclear membrane, that is the second. Complete disintegration of nuclear membrane mark the beginning of the transition to metaphase. In transition to metaphase, what will happen? So in transition to metaphase, the chromosomes are spread throughout the cytoplasm, and by this time the chromosome is maximum condensed. Then the kinetochore will get attached with the spindle fiber, and finally the formation of metaphase plate will happen. So if you see the sequence, it will be 3, 2, 4, 1, 6, 5. So just see, this is important question. Such questions are asked in which you are asked with the sequence of events. So from such kind of questions are asked from this chapter, so be very much prepared for such questions. Next question is, which of the following is characteristic of transition to metaphase? So first of all, understand where transition to metaphase comes. So we have prophase, and after prophase there is transition to metaphase, and then after this transition actually you will get metaphase. So basically this question is asking what are the characteristics of transition to metaphase. So during this phase, chromosomes are spread throughout the cytoplasm. Yes, it is correct. If you see the diagram in NCERT, they have clearly given the diagram for transition to metaphase. So chromosomes are spread throughout the cytoplasm. Chromosome arranged on a metaphasic plate. No, this is a characteristic of metaphase, not the transition to metaphase. No nuclear envelope. Yes, there will be no nuclear envelope, and spindle is attached to the kinetochore. Yes, this also happened during the transition to metaphase. So 1, 3 and 4. Third will be the right answer. Next question. How many of the following explain the metaphase plate? Metaphase plate, all chromosomes are arranged onto a single plane. Yes, metaphase plate arranged on the equator. Correct. Chromosomes have 2 chromatids. So yes, chromosomes have 2 chromatids during the metaphase. And in the metaphase plate, the spindle equator has all the kinetochore arranged on it. Yes, the kinetochores, all the kinetochores. So if this is the metaphase plate, right? So here all the this centromere, they are present onto the equatorial plate. So kinetochores are also present onto the equator. So 1, 2, 3 and 4, all of them are correct. Next question, if you see, during anaphase, what happened during anaphase? So here the centromere split one after the other. Does it split one after the other? Wrong. It happens simultaneously. 2 daughter chromatids are formed. This is correct. The daughter chromosome of the future daughter nuclei move to the opposite pole. Yes, the daughter chromosome of the future daughter nuclei, exactly NCERT line, they move to the opposite pole. So both 2 and 3 are correct. First one is wrong. Next question. During movement of chromatid during anaphase, select the correct statement. See, during anaphase, there are 2 poles. Now, in these two poles, when the chromosomes are moving, so this part is this is the centromere, right? And these are the arms. Now, on the basis of centromere and arms, question is asked here. So, centromere is directed toward the pole. As you can see, yes, it is directed toward the pole. Arms of the chromosome are at the leading edge. See, arms are they leading or they are lagging? So, they are not leading, they are lagging. So, it is wrong. Centromere is trailing behind. See, centromere is leading. No? Each pole receive half of the number of chromosomes. See, at each pole, equal number of chromosome goes. No, we know that during anaphase, the chromosome number get double. Right, so each chromosome get equal number of chromosomes. So this is also wrong. So only one is correct, rest are wrong. Which is the correct statement related to telophase? Here, chromosome lose individuality. During telophase, chromosome lose individuality. Yes. Centrosome appear clearly. Centrosome disappear. Nucleolus, ER, Golgi complex disappear. Wrong, they reappear. Two sets of chromosome present in one nucleus. No, they are not present in one nucleus. Two sets of chromosome are present in two nucleus, right? One set present here, one set present here. So this is also wrong. So the first one is correct, rest are wrong. Then which of the following do not happen during telophase? Centrosome cluster at opposite spindle fold, correct? The chromosome cluster at opposite spindle fold, correct? Chromosome present as discrete elements. During telophase, they are not present as a discrete element because the individuality of the chromosome is lost. Nucleolus reform. During telophase, the nucleolus reform, right? Yes. Each pole has one daughter nuclei. That is also correct. So third one is wrong, right? So second one don't happen during the telophase. Next. Which of the following related to cytokinesis? Furrow formed from the center to periphery. See, during cytokinesis, the furrow is formed from periphery to the center, not from center to the periphery. So this is wrong. Cell plate in the plant cell due to extensible wall, right? Cell plate is formed because of inextensible wall. So this is also wrong. In plant, new cell wall formation begin from the precursor middle lamella to form cell plate. Precursor, actually it is written opposite. Precursor cell plate to form middle lamella. So this is also wrong. Precursor is not the middle lamella. Precursor is cell plate which form middle lamella. In some cell, cytokinesis is followed by karyokinesis. Cytokinesis followed by karyokinesis? Yes. In some cells, cytokinesis is followed by karyokinesis. Here if you see, first what happened? Karyokinesis. And then we see cytokinesis. It is never that cytokinesis will be followed by karyokinesis. Always karyokinesis will be the first. Never cytokinesis happen first. So here, this is also wrong, right? In some cells, cytokinesis, so all of them are wrong here. All incorrect. All is incorrect. Then moving ahead, here, mitosis is equational division. Mitosis is equational division, it is correct. Mitosis is restricted only to diploid cell. Is mitosis restricted only to diploid cell? This is wrong. So statement 1 is correct, statement 2 is incorrect. So first one is correct here. Next one, mitosis can result in formation of. So again, it is related to haploid diploid. So read the statement carefully. First, haploid daughter cell with identical gene. So mitosis can, see here, can result in formation of. Yes, mitosis can happen in haploid. So haploid daughter cell with identical gene can be formed. Correct. Mitosis can result in formation of diploid daughter cell with identical gene. Yes, it can also lead to formation of diploid daughter cell. Haploid daughter cell with different gene. See, different gene is not possible. Diploid, different gene is not possible. So 1 and 2 is correct here. Next, which of the following show mitosis? Diploid microspore mother cell. Diploid microspore mother cell show meiosis. Haploid cell of lower plant. Yes. Haploid cell of social insect. Correct. Triploid primary endosperm cell. Do they show mitosis? Yes, they also show mitosis. So 2, 3 and 4 is correct here. Next, significance of mitosis can be. So here it is significance. So mitosis help in growth of unicellular organism, correct? Reproduction of unicellular organism. This is also correct. Growth of multicellular organism, correct? Reproduction of multicellular organism, correct? Increase in nucleocytoplasmic ratio. Now, see, in NCERT they have given, they help to maintain the nucleocytoplasmic ratio. So understand, if I plot a graph of nucleocytoplasmic ratio, then as the cell grow, the nucleocytoplasmic ratio decreases. But as the cell divide, the nucleocytoplasmic ratio increases. Then it will again decrease as it grow, and as it divide, it will again increase. So when the cell divide, the nucleocytoplasmic ratio increases. So this one is correct. Decrease in, so no, cell divide. So 1, 2, 3, 4 and 5 is correct, except 6 all are correct. Then mitosis help in, yes, it help in cell repair. Mature male gamete formation in plant, right? So mitosis help in mature male gamete formation in plant, yes, because microspore undergo mitosis to form mature male gamete, yes. So the pollen grain is formed now. So here you will see that mitosis is required, right? And before that you have meiosis. So mitosis and meiosis both are required in matured pollen grain formation or mature male gamete formation. Mitosis help in replacement of blood cell, correct? Growth of lateral cambium. Growth of lateral cambium is also through the mitosis, and mitosis help in continuous growth of plant. This is also correct. So all of them are correct. NCERT line are there. Which of the following statement about meiosis are correct? Meiosis ensure production of diploid phase in life cycle. Meiosis ensure production of diploid phase. No, it ensure production of haploid phase, because haploid microspore is formed. So this is wrong. Fertilization restore the haploid phase in the life cycle. It restore the diploid phase in the life cycle. Meiosis always lead to formation of haploid gamete. Yes, it lead to formation of haploid gamete. Meiosis take place during gametogenesis. This is also right. So 3 and 4 is correct here. Which of the following statement is true about meiosis? It involves 2 cycles of cell division and 1 cycle of nuclear division. Now in meiosis, what actually happened? Here 1 cycle of replication and 2 cycle of division. And here the division include nuclear division. So this statement is wrong. It involves a single cycle of DNA replication. Yes, replication occur once, so it is correct. It always take place in a specialized diploid cell. Yes, it is also correct. It always take place in a diploid cell. It involves crossing over between non-homologous chromosome of sister chromatid. Now this is wrong. It should be homologous chromosome of non-sister chromatid. Right, so this is also wrong. So correct is 2 and 3. Next question. Which of the following is the longest? Very simple question. Prophase, right? Even if you have to compare prophase 1, prophase and prophase 2, so prophase is the longest phase. Then longest and the shortest. So longest we have just said, prophase is the longest and shortest one is the anaphase. So second one will be the right answer. Longest is prophase and shortest is anaphase. The prophase 1 is divided in 5 phases based on. Now this is again from the NCERT line. Clearly they have given it is based on chromosome behavior. Next one, stage which can last for months to year. Stage which can last from month to year, diplotin, right? Oocytes of female. In human reproduction you must have studied. In diplotin can last for months to year. Enzyme mediated process. Which step is an enzyme mediated process? So we know that crossing over need which enzyme? Recombinase, right? And recombinase enzyme is involved in which phase? Where crossing over happens. So where crossing over happen? In the pachytene. Then chromosome become distinctly visible. So distinctly visible in leptotin, right? Then electron micrograph show complex structure. Here which complex structure it is talking about? So it is talking about synaptonymal complex. So this synaptonymal complex is found in zygote. And lastly, chiasmata disappear. So chiasmata will disappear in dikinesis, right? So correct will be If we see, one will go with A, 2 is going with D, right? 3 is going with B, right? 4 is going with C. So last, fourth one is the right answer. Then moving ahead, by the end of dash during meiosis, nucleolus disappear and nuclear membrane breaks down. By the end of which phase? Right? So it is talking about the prophase, right? Prophase 1 of meiosis, which stage? So it is talking about dikinesis, right? Dikinesis. By the end of dikinesis, nucleolus disappear and nuclear membrane breaks down. Ask that which of the following is incorrect about metaphase 1. So bivalent chromosome are aligned onto the equatorial plate, correct? Each chromosome has 2 chromatids. This is also correct. Spindle get attached to the kinetochore of sister chromatid. Now here if you see, because the chromosomes are present as a bivalent, so here the spindle get attached to 2 different centromeres. So this one is wrong. Chromosome are maximum condensed in this stage. This is correct. So here third one is incorrect. Next one. Which of the following is correct about anaphase 1? During anaphase 1, each chromosome has 1 chromatid. This is wrong. During anaphase, anaphase 1, each chromosome has 2 chromatid. The centromere split. This is also wrong. The sister chromatid separate. Each chromosome has 4 arms. Yes, chromosome has 4 arms because it has 2 chromatid. The number of chromosomes get doubled during anaphase 1. This is also correct. So, anaphase 1, the 3 and 4 both are correct here. Next question. Which of the following is correct about telophase 1? During telophase 1, during this phase, cells are called dyad of cell before cytokinesis. See, not before cytokinesis, it is after cytokinesis. It is called dyad of cell. Nuclear membrane and nucleolus reappear during telophase, correct. Chromosome undergo complete dispersion. Wrong. Chromosome don't undergo complete dispersion. NCERT line. Meiosis end with this phase. No, meiosis don't end with this phase. Still there is meiosis 2 left. So correct answer is 2. Next one. Which of the following is correct about interkinesis? We know that interkinesis is the stage between meiosis 1 and meiosis 2. This phase is called interkinesis. Now, we know that this is a short phase, right? Here, no DNA replication happened. No DNA replication, right? And here, only some protein synthesis happened. Protein synthesis happened. So, on that basis, let's answer this. So, interkinesis. It is similar to interphase. No, it is not similar to interphase. DNA replication take place. No, DNA replication don't take place. Protein synthesis occur. This is correct. It is a long phase between meiosis 1 and meiosis 2. No, it is a short phase between meiosis 1 and meiosis 2. So third is the correct answer. Next one. Read the following statement and choose the correct. Okay. Prophase 2 is simpler than prophase 1. Yeah, correct. Anaphase 2 has half the number of chromosomes as compared to anaphase 1. It is wrong. Both anaphase 1 and anaphase 2 both have equal number of chromosomes, that is 2n. Both will have equal number of chromosomes. Number of chromosomes in a dyad of cell and tetrad of cell is the same. Yes, it is also correct. Dyad also have 2n and tetrad also have 2n. Each chromosome has only 1 chromatid during metaphase 2. During metaphase 2, if you recall, each chromosome have 2 chromatid, not 1 chromatid. So this is also wrong. So correct answer will be 1 and 3. Next one. Meiosis take place only during sexual reproduction. Correct? Meiosis take place during sexual reproduction. Mitosis not required during sexual reproduction. This is wrong. Mitosis is also required during sexual reproduction. Meiosis always takes place in diploid cell. This is correct. Mitosis sometimes take place in haploid cell. Yes, we have seen mitosis taking place in haploid. Mitosis conserve the same number of chromosomes from one generation to next generation. See, mitosis is called equational division because daughter cells have same number of chromosomes. But from one generation to next generation, it is not maintained by mitosis, it is by meiosis, right? It is by meiosis. So that's why this statement is wrong. So 1, 3 and 4 is correct. 1, 3 and 4 is correct. So here, okay, so just consider this and 1, 3 and 4. So mind it, 1, 3 and 4 is correct here. Next one. If a plant leaf cell has 40 chromosomes, now we are entering into some numerical type question based on this. If plant leaf has 40 chromosome, number of DNA molecule during metaphase and anaphase. See, if plant leaf cell has 40 chromosome, means what I am telling, 2n is equals to 40, right? So it is asking about number of DNA molecule during metaphase and anaphase. Now, so if 2n is 40, what will be the number of chromosome during metaphase? So during metaphase, it will have 40 chromosome. Now, if it has 40 chromosome, so each chromosome has 2 chromatid, right? Each chromosome has 2 chromatid, and we know that 1 chromatid is equals to 1 DNA molecule. In the beginning, we have seen, right? So here how many chromatid? So each chromosome has 2, so 40 into 2, that is 80 chromatids, right? So 80 chromatids, so here there will be 80 DNA molecule. So in metaphase there is 80 DNA molecule. Now what in anaphase? So see, here in anaphase the chromosome number get double, so the number of chromosome will be 80, but the difference here will be each chromosome only have 1 chromatid, right? Because splitting of this will happen in anaphase. So each chromosome, so 80, and because each chromosome has 1 chromatid, so 80 into 1, so there is 80 chromatid. So see, here also 80 chromatid, here also, so here also there will be 80 DNA molecule, right? So overall 80 and 80 will be right answer. Next question. Here, cell has 80 chromosome in G1 phase, right? So in G1 phase, we know that the number of chromosome is 2n and 2n is 80. Now number of chromosome by the end of S phase, metaphase and anaphase. It is asking about number of chromosome. Now see, if G1 phase has this, so by the end of S phase you will have 2n still will be there, G2 will be 2n, right? In prophase also 2n, metaphase also 2n, anaphase, we know that it get doubles chromosome number, so it becomes 4n, telophase 4n, and then after cytokinesis we get again 2n. So now on that basis you can simply say S phase 2n, so it remain 80. Then it is asking metaphase, so metaphase also 2n, so it is 80, and finally anaphase, so anaphase is 4n, so it will be 160. So it is 80, 80, 160. See, this table you can use to find out for all the stages, right? You get the number and then accordingly you can calculate. Next one, number of chromosome at G1 of meiosis is 12. So again at G1 2n is equals to 12. Now it is asking number of chromosome during. So these are the stages of meiosis. Now in meiosis, anaphase 1. So in anaphase 1 of meiosis, the chromosome number don't get double. So if for meiosis, if I write the table, so here G1 is 2n, S again 2n, then G2 2n, then prophase 1 2n, then metaphase 1 2n, anaphase 1. See, it doesn't become 4n, chromosome number don't get double, so 2n, then after anaphase telophase 1. So in telophase 1 it become n. So this also represent the diad. Following that, in prophase 2 it is 2n, sorry, it will remain n, no, it will be n. Then metaphase 2 it will be n. Now anaphase 2 will become 2n. Why? Because here splitting of centromere will happen, and by the end of telophase 2 you will have n number of chromosome. Means here it is represented by diad, it is represented by tetrad. That's why we say diad and tetrad both have n. Now on this basis we can easily find out this. So now if 2n is equals to 12, so in anaphase 1, you see anaphase 1 2n, so it will be 12. Metaphase 2, see metaphase 2 is n, right? So it will be 6. Anaphase 2 2n, so it will be 12. Diad of cell will have n means 6, tetrad of cell also n means 6, right? So 12 6 12 6 6. See which one is the answer? 12 6 12 6 6. So second is the right answer. Again, this table is very, very important. It will help you to solve all such questions. Then 4 microspore mother cell after meiosis will form how many cells? See, we know that microspore mother cell undergo meiosis, and we know that one meiosis produce how many daughter cells? So one meiosis produce 4 daughter cells, right? 4 daughter cells. So here when we say 4, so 4 into 4. So that is 16 is the right answer. Then here if we see further, one cell after 4 mitosis form how many cells? So see here, for mitosis the formula is simple. If it says after 4 mitosis, so the formula is 2 to the power n. So final number of cell, this is equals to 2 to the power n. Now n is the number of mitosis, so here n value, n is equals to 4, so number of cell will be 2 to the power 4, right? So it will be 16, right? It will produce 16. Such simple formula, remember, 2 to the power n. Then moving to the next one, number of chromosome at G1 is 18. Again, G1 is equals to, so it is 2n, which is equals to 18. Now it appears similar, but yeah, it here some twisted. It's mixture of mitosis and meiosis. So it is asking that at each pole of anaphase 1. See, we know that in anaphase, just before we have seen that in anaphase 1 the number of chromosome is 2n. It doesn't increase, right? But at each pole you will get n and n. That's why total is 2n, right? So at each pole it is n. 2n is 18, so n is equals to 9. At each pole of anaphase of mitosis. Now you see here in mitosis anaphase we have seen that it become 4n, and in that case, in case of mitosis, both side will get, because 4n, so 2n on this side and 2n on this side. So that's why at each pole of anaphase will be 18. At metaphase of mitosis, right? So in metaphase we know that the number of chromosome is 2n, right? So 2n is 18, so it will be 18. And lastly, number of tetrad during prophase 1. Now here it is important to know this relation of tetrad. See, there are two terms. One is called bivalent and another is tetrad. So remember, one bivalent is equals to one tetrad is equals to one pair of chromosome. One pair of chromosome, right? So here it is asking number of tetrad. So if we know the number of pair of chromosome, we will say that it is tetrad. So here if I ask how many pair, so 18 chromosomes, so there will be 9 pairs, right? So 9 pairs, so 9 tetrad. Now this relation you remember. Question is asked from here, so the answer here will be 9. So it will be 9, 18, 18, 9. Let's see which one is. So third is the right answer over here. Now next question. Next question is about this. So if we see the next question, it says if the pea plant has 14 chromosome, now number of bivalent, so this is the relation. Now bivalent, tetrad and DNA molecule. So here just again I will repeat the relation. One pair, right, is equals to one bivalent, and one bivalent represent one tetrad, right? Now during this stage, because each chromosome has 2 chromatid, so one chromosome will have 2 DNA. So on that basis let's answer it. So 14 chromosome means how many pair? 7 pair. So how many bivalent? 7 bivalent. How many tetrad? So 7 tetrad. And how many DNA molecule? So see, there are total how many chromosome? So there are total 14 chromosome, right? Because there are 14 chromosome and each chromosome has 2 chromatid. So number of DNA molecule here will be, okay, 7 tetrad, so 14 chromosome, so it will be 14 into, because 14 chromosome and each chromosome has 2 chromatid, so it will be 28 DNA molecules. 28 DNA molecules. So number of DNA molecules will be 28 on the basis of number of chromosome. So here if we see this, I think none of it is right. So here I think this is right, but 14 should be 28. Then it will be right. Right? Now next one. During the prophase of mitosis, each cell has how many aster, how many centrosome, centriole and spindle fiber. So in one last question we have seen that, but let me again draw it. See, during prophase, right, there are 2 poles and at each pole we have 1 centromere, and we know that 1 centrosome, and this centrosome is made up of 2 centrioles. As you can see, on both the pole we have 2 centrosomes. So this is centrosome 1, right? Centrosome 1. This is centrosome 2. And both the centrosome have 2 centrioles. We can say that. And these centrioles are perpendicular to each other. 2 centrioles, right? Now, these centrosome will have radiating microtubules like this, right? They have radiating microtubules. So I will label this as radiating microtubule. And here also there will be radiating microtubules. Now this radiating microtubules along with this centrosome, this is called aster. So can we call it aster 1 and this one is aster 2. So in a dividing cell during mitosis, how many aster are there? 2 aster are there, one at each pole. Then along with this aster, there are some spindle fiber like this, spindle fiber. Spindle fiber, right? So here the anther along with the spindle fiber together is called mitotic apparatus. So the whole thing is representing one mitotic apparatus. On this basis, let's see this question. So during prophase of mitosis, each cell has how many aster? 2 aster, as you can see here. Each cell has how many centrosome? So you can say 2 centrosome. How many centriole? So each centrosome has 2 centrioles, so to this side, to this side. So there are total 4 centriole. And how many spindle fiber are there or mitotic apparatus are there? Not spindle fiber, how many mitotic apparatus are there? So overall, it represents 1. So you can see 2, 2, 4, 1. So first is the right answer over here. So this was the all question related to this. It was a very important chapter, so revise all these questions. Now if you have liked this video, please share it with all your friends, right? And if you want the PDF of this these questions, if you want to PDF of this, so below is the link where you can go and download the PDF and you can solve it for your revision purpose. So do like this video and keep on connected. I will keep on sharing such all chapter wise MCQs. So best of luck, continue working hard.